Yeah, thanks very much. Well, let me first introduce the other people behind the table. So Melissa Little works on the development of kidneys from stem cells. Guo-li has published on using mini brains to study viral infections, Zika virus in particular. Jürgen Knoplich from Vienna has been the person who, I guess, developed the mini brain technology to make brain tissue from stem cells. And I'll start with describing the field, and I'll give you some examples of what we have done in the Netherlands. So we're talking about organoids. What are they? They are small structures that we grow from stem cells, and they organize themselves into structures that really start looking like bits of real organs. They're not entire kidneys or lungs or guts. They're very small, yet they have all the critical characteristics of what you would hope to study in real life. And we can make them from human stem cells. So rather than studying everything in animals, we can actually use human stem cells and study disease processes, drug activities, et cetera, on these mini versions of the organoids. There are two types. I'll be talking about one type. So this starts from adult stem cells. In this slide, you'll see that we believe that every organ, as we sit here, has its own stem cells. So they're really very dedicated stem cells. Lung stem cells can make lung tissue. Bone marrow stem cells can make blood cells. They have been used for 60 years, but they cannot make something. So bone marrow stem cells we don't believe can make kidney or can make lung. So these are called adult stem cells or pluripotent stem cells. There's a second version of stem cells, embryonic stem cells, or the synthetic version, the induced pluripotent cells or the iPS cells, and they in principle can make every part of our body, and I guess the three other people will give examples of how you can actually use embryonic stem cells or iPS cells to build entire human tissues. This little movie shows you what they look like when we make mini guts, so you don't really immediately recognize a gut in these structures, but as you can see, they're extremely vigorously growing. And when you look at these things with microscopes, you'll see that all of the cell types, all of the aspects of what you would normally see on the insides of the gut is represented into these mini structures. You have to realize they're probably 1 to 2 millimeters in size at best. There are no blood vessels yet. People are trying to engineer them in, so they cannot grow beyond that little size. This can be done for human gut. So essentially you take a very small biopsy, a little bit of tissue from a patient or from a healthy donor, and then you can propagate it in the lab forever. You can send it around the world. You can test drugs. You can infect it with viruses, and I'll give some examples. You can freeze them, thaw them, and anything we've learned over the years to do with normal cell lines, looks like we can do this with these organoids. And they're really, if you take them from a healthy donor, they are healthy cells. You can give them back to patients, or to mice in this case, and they will make normal tissue. Now, there are now protocols developed by us in Amsterdam, but also by many other labs, that was a whole list of human organs that we can now grow, and other labs can grow straight from a little bit of tissue from a patient. And this includes the stomach, the small intestine, the colon, liver, pancreas, lung, breast, and various other parts of our body. Importantly, we can do this from healthy tissue, and then when we do this correctly, it's healthy tissue, even in culture, even after a year. But also you can do this from disease tissue, for instance, from hereditary disease patients. I give one example, but you can also do it, for instance, from cancers. So you can grow cancers in the lab and study various aspects of them. And again, I'll get back to that. Now, this movie doesn't work, but what it had shown is that in principle you can grow mini organs from healthy stem cells, and they make healthy tissues, and you can transplant them back. So in the long run, and we're talking many years, this will probably be applicable in humans and might replace some of the organ transplant approaches. So you, rather than having to wait for a traffic accident victim to have a kidney or a lung or a heart, this technology offers that you can actually have a healthy donor, like a blood donor, donates a bit of tissue, goes back home, the tissue is expanded, frozen, and then when needed, you can give it to a patient. We can do it in mice, various labs around the world, but it's a far way away before we're allowed to do this and before we can really be sure that it's safe to do this in humans. So that's regenerative medicine purposes. Another early one, particularly for the technology with adult stem cells, is, how do I... How do I stop the... Oh, this is a Dutch translation while I'm... So you can actually take a sample of a tumor, and you can take a sample of normal tissue directly next to the tumor. You can grow it in the lab, and then use it as an avatar for the patient, for the tumor. And here you see what they look like under the microscope. You can actually then analyze the DNA of the tumor, and as you might know, tumors have small mistakes in their genomic code, and this causes them to be malignant. But that you can do directly on the tumor as well. But the fact that you can now grow them outside the patient in the lab, and this takes a few weeks, you can now, with robots, screen large numbers of drugs and see which one will kill the tumor cell. And in the end, so we hope to be able to use this technology to actually predict for any given cancer patient whether, what is the best individual drug for that cancer patient. And these organoids are then, they act like avatars of the patient. And rather than exposing a patient to a single drug and see if it works, you can expose the avatar of the patient to 10, 20, 100 drugs and then find what's best. This we are currently testing and many other labs around the world, but the next one is in place for cystic fibrosis, where Vertex has developed two miraculous drugs for cystic fibrosis, but it's very unpredictable what patients will benefit. And they're rare versions of the disease where trials are impossible because the numbers of patients are too low and also the business models don't work for companies. But it turns out that on a tiny bit of tissue, lung, but also colon or kidney, you can, and this is the test, I won't explain it, but you can actually show that a patient has cystic fibrosis, and you can then expose the mini-gut or the mini-lung from that cystic fibrosis patient to these extremely expensive drugs of Vertex and decide in the lab what would be a good drug. And actually you can repair the gene. That's what you see here by CRISPR-Cas9 also. It's not so relevant here. And actually in Holland now we have a situation in place where every time we recognize a patient, and this was our very first one, Fabian, who appeared in the most popular Dutch talk show and told his story. So he has a very rare version of the disease, has not been in trials. The Vertex drug cannot be given to him. At least it will not be paid for by the insurance companies. And the drugs probably cost around 300,000 or 400,000 dollars per year. So we found that he reacted very well in an organoid test. Based on this, he was given the drug and he miraculously recovered in hours, actually. And he's now back on the hockey fields. And based on this, now we have a number of patients that get this drug, and we're currently setting up a program with the insurance companies to screen all 1,500 patients in Holland with CF, create a biobank, so find the patients for which these drugs work, and then there will probably be many, many left for which the drugs don't work, but we know that Vertex, as well as several other companies, are developing new drugs, and what we see in the lab already is that these new drugs treat a different spectrum of patients. So eventually we hope to be able to link every individual patient to his or her best drug. And that's, I guess.
Can I just ask one quick question?
Yeah.
The gene of the patient, whatever mutation they have, determine whether they're going to benefit? Like, why do you need to?
Yeah, we'll have a major the question session after this, but I can give you a very brief answer. So the drugs are developed for individual mutations. One of them is very common, the delta 508, so that's what Vertex has been using as their target mutation. But it turns out that even amongst that group, probably half of them respond well, others don't. So there's other genetic influences that are unpredictable, that doesn't allow you to predict whether the patient will or will not. But that's actually the group where all patients have the right to the drug. But then there's another 50% that have, rare or extremely rare versions of the disease, of the mutation, where Vertex cannot test because there are not enough patients to do trials. And that's where we think this NS1 approach works very well. Fabian is an NS2 actually, because his aunt had the same mutation, and they both do very well on this drug. Okay, so I think we'll move over to Jürgen, who will talk about the other type of technology. So this is now the iPS-based technology to build little bits of human brain in a dish.
Okay, so my name is Jürgen Knoblich. I'm a scientist at the Institute of Molecular Biotechnology of the Austrian Academy of Sciences in Vienna. And what I would like to do in my presentation is tell you about fairly different technology that can also be used to generate organoids. Now, what we've heard from Hans Clevers was one specific way of generating organoids, where you start with adult stem cells that are derived from patients to generate various types of organs, which then can be used for disease modeling, drug testing, and eventually maybe regenerative therapies. Now, this type of technology works for any organ that has a rapid turnover in our body, because it basically uses the knowledge that we have about tissue regeneration to generate tissue in the laboratory. Now, this can be applied to a number of tissues, but there's one tissue where it certainly cannot be applied to, and this is the human brain. And so what I will be telling you about is the work from my lab that is mainly concerned with generating human brain tissue in the laboratory. So the way we do this is we recapitulate the development of an organ. We start with pluripotent stem cells. Ever since the groundbreaking work of Shinya Yamanaka, we can generate pluripotent stem cells, so-called iPS cells, from any human individual, whether healthy or diseased. And then what we do in the lab, and this is really the fundamental principle of this type of organoid research, is we recapitulate human development in a laboratory. And so I want to emphasize that all of these technologies rely on a very large body of previous work that was done by developmental biologists, and that has identified all the individual factors, so-called morphogens, that tell the cells whether they form anterior head tissue or posterior trunk tissue or dorsal or ventral or any different part of the human brain. And what we basically do is nothing more than just going into this literature and trying to recapitulate these developmental processes that have been identified by others before. And so the basic principle and also the hope of this type of technology is that one would start with any human individual, use reprogramming to actually generate pluripotent stem cells, which we then either with certain cell culture, tissue culture, media, or with certain growth factors drive into a certain tissue fate, and through miraculous events that actually also have been found already in the mid-19th century, when we drive the cells into a particular tissue fate, they are then able to organize themselves, to basically recapitulate the three-dimensional organization of that tissue. Eventually, we then end up with structures that we call organoids. And although our organoids and the ones that Hans Clevers has been describing are both called organoids, they're actually something quite different, both in the level of complexity as well as in the way they are actually developed. And the hope is that we then will be using those organoids, number one, to understand human disease. The reason for this is because so far we have mostly been using laboratory animal models, but certain diseases, particularly the ones that affect the brain, cannot easily be modeled in laboratory animals. We then can use them to test drugs. The major intention here is to save animal experiments, but also to do these experiments in a higher level of precision, and ultimately also do organ replacement, but I think that's far away from the brain. So the methodology that my laboratory has been developing is illustrated here. So basically what we have done is we have been able to generate organoids that to some degree resemble the human brain in culture. So for this we start with human pluripotent stem cells, typically iPS cells, which we dissociate and then rapidly reaggregate at the bottom of a tissue culture well. And we do this under conditions where those pluripotent stem cells no longer remain pluripotent, and they start to go into development. And the very first event that happens during development is the formation of the 3 germ layers, ectoderm, mesoderm, and endoderm. And that's what these cells then will do. And then by simply changing the cell culture media, what we're getting are these balls of neuroectoderm. Neuroectoderm is the part of a human embryo that will later give rise to the central nervous system. And these balls of neuroectoderm are then embedded in a substance that's a little bit like collagen or like a jelly, and this is called matrigel. And matrigel really is the holy grail of organoid technology. For some reason, it seems to have all the factors that are needed for generating organs in vitro. And the reason for that is because it very much resembles the composition of the material that's in between cells in a human embryo. And so we embed them in these droplets of matrigel, where we then culture them initially just in a tissue culture plate, and later either in a device that is called a spinning bioreactor or just on an orbital shaker. And over time, we then see the development of very complex structures. So these are 2 examples of what we call cerebral organoids. So here you see the development of an actual human cortex. This lumen here is the so-called lateral ventricle, which is a hallmark of mammalian brain development. Here is a second cortex, and here is a third one. Over here is another example. There's a cortex here, and there's one here. And here you actually can see the development of a human eye. We have made cross-sections through these eyes, and the way the cells are layered very much resembles the way this looks in an actual human embryonic eye. This is a cross-section through a cerebral organoid, so there's a cortex here. What I want you to note is that the layered structure of this developing cortex very much resembles that of an actual brain. So in red are the stem cells, and these are the cells that are actively dividing, and they divide in a way such that they generate both stem cells, but also differentiating active neurons. And these neurons will then migrate out here, and here they will start to interact with each other, to send out axon cables and to communicate with each other. And all these processes can be recapitulated in this cell culture tissue. So if I would show you a cross-section through an actual human embryo at this stage, it would look indistinguishable from this. Down here are other areas of the brain. So this is the so-called choroid plexus, which is the area of our brain that generates what is called the cerebrospinal fluid. And here are other brain areas. I want you to know that they look different. They're organized in a different way. So our organoid system recapitulates various different areas, various different parts of the developing human brain. And we also know that the neurons that are forming in these organoids are electrically active. They communicate with each other. They do not process any information. It's more like a random communication, but the neurons are active, and that actually increases the number of things we can do with those organoids. We had in the past already used this system to model a human neurodevelopmental disorder. This disorder is called microcephaly and results in a severely reduced brain size. We took pluripotent stem cells, iPS cells, or we generated iPS cells from a patient who suffers from this disease, and indeed the organoids were much smaller, and this allowed us to then actually go back in history and ask why in this patient is the brain so much smaller, and we could come up with a plausible mechanism. What we could also do is we could repair a mutation that we found in the genome of that patient, and then the brain size and the early defects that led to the reduced brain size actually came back. Now, all of this work has been published a couple of years ago, but what I would also like to do in the end of my presentation is to come to what I actually presented at this conference. These are fairly new data that actually have allowed us to significantly improve our experimental system. And the reason why we need to do this is because, of course, ultimately we would like to use our culture system to not only model very severe neurodevelopmental disorders like microcephaly, but also the more common and more widespread disorders like epilepsy or autism, and eventually maybe even schizophrenia. But there's one problem with the organoid system which we needed to overcome for this. So in this example, you see that there's a nice stereotyped arrangement of the various types of the human brain in the organoid, but in a way, actually, our organoids look more like this. So this is a normal car. This is how it looks in the brain. Everything has its own place. The forebrain is always here, cerebellum always here. But in an organoid, the various parts of the brain are actually randomly arranged. So it's a bit like a car where you would have the wheels on top, the seats in the back, and the exhaust coming out at the top. And so much of our research now is actually directed towards putting some order into this. And in my presentation here, I presented two recent developments in my laboratory. So on this slide, I just show you two typical examples. So this is one organoid, this is another organoid, and you can see that they just look very, very different. Although both of them actually have the right structures, but they're very arranged in a very different way. And so we've taken two distinct approaches to make this more reproducible. The first one is we combine organoids with bioengineering. So we collaborate with bioengineers who generate what are biocompatible fibers, and that allows us to actually change the structure of the organoid, and I showed that this actually can improve how the organoids look. And what we're currently working on is we're trying to modify these fibers in a way so that they recapitulate endogenous anterior, posterior, or dorsoventral axis of the brain. And the second approach that we took towards this was we generate the different parts of the human brain separately. So, for example, we make one area of the brain in one culture, and we make another area of the brain in another culture, and then we put them together to generate an organoid that actually has a predefined axis, in this case a dorsoventral axis, and we could show that we can then actually see both the axons, the cables going from one into the other part of the brain, and that we can also see neuronal cells migrating from one part into the other part. And what we're doing right now is we're generating iPS cells from patients where we suspect that these events are affected, and we hope that this will allow us to actually get at the more disease-relevant processes or widely disease-relevant processes to eventually come up with organoid models for some of the widespread neuropsychiatric disorders. Thank you.
Okay, so good morning. My name is Guo-li. I'm a professor in the Department of Neuroscience at University of Pennsylvania School of Medicine. So the work in my lab is essentially quite similar to what Jürgen just described to you, is trying to generate brain organoids and use this organoids as a model system to study neurodevelopment and to study developmental diseases. So this has been introduced by Jürgen as well, that we can generate pluripotent stem cells from individual, either healthy individuals or people with diseases. And normally, by learning a lot of previous studies, we can show that these cells are pluripotent and can be differentiated into every single cell type in our body. This has been mentioned both by Hans and Jürgen, and we've been doing this using 2D differentiated cultures, which is a single layer of cells. At bottom of the dish, we can differentiate the cells into neurons, for example, the nerve cells in our brain, or glial cells in our brain. Just as Jürgen just introduced to you, an innovative research from his lab have developed approach to differentiate these pluripotent stem cells into a 3D structure, much like the brain, using a spinning bioreactor. So we follow this and trying to also develop the same strategy to generate brain organoids. And the commercial available spinning bioreactor is shown at the bottom of this incubator that we commonly use in the lab. And as showing here, it's not, the pointer is not showing here. At the bottom is a commonly used, commercially available spinning bioreactor. You can see it's like a beaker size, you have to use a lot of media, so it's associated with high cost. And also Jürgen mentioned a little bit that organoids grow under these conditions. If you completely rely on intrinsic properties to grow into cerebral organoids, it has some high variability and not really truly organized as a normal brain. So first, to deal with the high cost, so we try to come up with a strategy to miniaturize the spinning bioreactor. So this as a summer project for several high school students, we challenged them to generate a miniaturized spinning bioreactor that will much reduce the cost by reducing the volume that you need to in this bioreactor. So this is a design from a 3D design by these high school students, and you can see instead of the magnetic-based spinning bioreactors that are commercially available, they decided to switch to a motor-driven gears. So the gears are connected to each other. In this, you can see there's a 3x4, so an array of gears that are connected to each other. So if you just spin the main gear, which is taller at the up right corner, all the spinners will spin, and this driven the spin of the blades at the bottom, which directly stick into a 12-well culture plate, which is only like a bottle cap size. So the miniaturized spinning bioreactor will cut the cost at least 20 times less. And using this, by the way, also, after the 3D design, we actually use the 3D printer to print out individual pieces, and they assemble this together. Once you assemble them, they become a lid. So it's very easy to use. You just take it off, put it back on. And use this, instead of generating a whole cerebral organoids, we decided maybe we should reduce the complexity by generating a brain region-specific organoids. In this case, I'm showing you an example here. We can generate forebrain-specific organoids. And I'm just going to tell you what we have. Since this is already published, we can show that this organoid is brain region specific. It's in forebrain identity, where the brain area that is mostly evolutionarily enlarged part in our brain that is responsible for all the high functioning of our brain. And showing example here, we can have human-specific types of neural stem cells in these organoids. In addition, we can also see neurons of all 6 cortical layers. We know that our cortex is stratified with 6 layers, and we can identify neurons with all 6 cortical layers in our organoids. What can we do to use these organoids? So one of the things we do is using these organoids to study recent outbreak and the suspected link between Zika virus and microcephaly. Jürgen already introduced to you that using these organoids, they can study genetic forms of microcephaly. And I'm trying to show you that using organoids, we can also model disease-induced microcephaly. We started with 2D monolayer cultures and identified a specific type of cells, which is the neural stem cells that is highly susceptible to Zika viral infection, a population which is also highly affected in genetic forms of microcephaly. And then we use our 3D brain organoids, the forebrain-specific organoids, in culture to see what's the impact after Zika viral infection. Again, it's showing beautifully nicely at the left, in the middle panel, that's the control. You can see the ventricle, very well organized the stem cell layers, as well as the cortical plate layers. However, if you see at the right panel, after Zika infection, you see a collapsed organoids, as well as reduced numbers of both neural stem cells, as well as reduce the numbers of neurons generated. So these are the features much like microcephaly found in patients. A lot of the fetus with Zika infection exhibit collapsed cortical tissues and wrinkling, including the collapsed skulls as well. And so this is the final summarizing slide to show you what we are. And I just introduced you with the forebrain organoids that we generated. In addition to that, we also generated from these pluripotent stem cells, we also generated organoids from midbrain, also from hypothalamus origin. And with the help of those high school students, not only they developed this single layer, single plate format spin, miniaturized spinning bioreactor. Now they have generated this tower version of miniaturized spinning bioreactor where you can spin 100 wells or at the same time, all with the same speed because they are all driven by a single gear initially. And I just show you an example. We can use this system to model disease, for example, Zika-induced microcephaly, and what is next and what's happening in the lab recently. First, we have already done to try to identify drugs that are protective or antiviral after Zika infection using the 2D cells types as well as 3D cerebral brain organoids. And we have done an initial drug repurposing screen with FDA-approved drugs or the drugs that is in the clinical trials right now. And we have identified 2 classes of drugs. One is antiviral, and the other one is neuroprotective. Currently, we are conducting a much larger drug screen with 20,000 small molecules. And another area we are working on is trying to develop a point-of-care diagnostics. We know that most of the diagnostics now is based on PCR, and that disappeared in patient quite rapidly after the infection, usually disappear within a week. But we are trying to develop approaches for diagnostics that based on proteins, so that will last much longer, and we can retrospectively detect the infection. Of course, one question that we are really interested in is to use this brain organoids or mini brains to study the basic biology of organ generation with a focus in the brain. So that's it.
Okay. Yeah, and maybe I should add that actually, so the outbreak of Zika is 2 years ago, and it was noted that lots of babies were born with small heads, but the link with the virus was not, nobody knew whether the virus is really causing this. And I think 1 in 20 of the babies have this, of the infected babies. So I think this was a breakthrough that actually showed that the virus causes microcephaly. Right? And there was a few other labs, but we, Jürgen and me, have decided that your paper was the best one.
Thank you for this.
Okay, switching gears from brains to kidney. I work on making kidney organoids from pluripotent stem cells. So these are human pluripotent cells, as you've heard before. And what you're looking at on the left here is actually the nephrons of a forming kidney in a dish. So we've long been interested in the kidney. Renal failure is actually a disease that's growing at about 6% per annum worldwide, and the treatment of kidney disease really hasn't changed for more than 70 years. If you're very lucky, you get a transplant. Most people with end-stage renal disease will have to live with dialysis, which is extremely poor treatment in that it only provides about 10% renal function, and it has actually a very high mortality rate. And so we were interested in the long term in whether we can do something to find an alternative to dialysis. And really, as you've heard, what we're doing is to take human pluripotent stem cells and see if we can recreate kidney tissue in the dish. So what we've discussed to date is around brain organoids. Does that work? No. Okay. I won't touch it. What you can see here is the general approach, and that is that you can take essentially any adult somatic cell. So I've got fibroblast up here, but as you know, you can actually reprogram any cell, and that means you can start with any individual, and you can go down to the pluripotent stage and form an organoid. And you can see Madeline Lancaster and Jürgen Noblich's early brain on the left, which we've just heard about, and the eye. And what we set out to do was to try and find a protocol to make a kidney. And again, as Jürgen has explained, this really relies upon a long-term understanding of how the kidney normally forms during development. And so we've actually developed a protocol where we can start with pluripotent stem cells and then slowly walk them through early stages of development into what is the intermediate mesoderm. So this is very different to brain. We actually have to pattern to a completely different cell type. And we actually then take all of the cells in those dishes, and instead of putting them in a spinner flask, we actually put them on top of a floating membrane, which you can see in the middle there from day 10. And gradually in the dish, the cells actually self-organize, and they start to form epithelial structures, which are the early nephrons, and those nephrons start to elongate and pattern, as we would expect would occur in a developing kidney. And so by the end of this process, what you can see on the left here in this movie is actually a scan from the bottom to the top of one of these, what we call organoids. And at the bottom you can see a branching structure, which is the collecting ducts in yellow that connects up in red to the tubular epithelium, and then these balls of green at the top are the glomerulus, or the forming filter of the nephron. So normally during development, that filter will be the part that starts to filter the blood, and then the urine will pass down the tubules. So on the right-hand side, you can actually see from the pink, walking up through green to blue to red, is all of the parts of the nephron we would expect to be there in the kidney. We have a collecting duct, a distal tubule, a proximal tubule, and then a glomerulus. Now, I'd just point out again that this started as a homogeneous cell culture of pluripotent stem cells, and it's essentially patterned and self-organized into this very complex structure. In fact, surrounding these nephrons, there's an interstitial population, and we also have vasculature forming within the organoids themselves. So this was a very big breakthrough in terms of kidney biology. The kidney is a very complex organ, and so to get this amount of cellular diversity was quite amazing. And the question really is, how do we move from organoid to application? And just as with the brain organoids, we can project that we may be able to use these for disease modeling, we may be able to use these as model of human tissue to screen for drugs, and in the long term, we may be able to use the cells that we grow in the dish for either cellular therapy or for bioengineering replacement tissue to go back and feed the patient. The patient on the left-hand side can be the source of the cells because it's possible to take any cell from the patient and turn it into a pluripotent stem cell. And so this is a loop that we hope we can close. We're a long way away from this. I've got another arrow in here which I think is quite important. This does give us a model in a dish that allows us to understand human development. These are obviously not points of development that we can experiment on, but by recreating in the dish, we believe we'll begin to understand some of the early events in human that are undoubtedly different to development in an early mouse or other model animal. So I want to just talk briefly about disease modeling. So as I've said, you can start with any person on the left-hand side, and so we're actually starting to use these to model inherited forms of kidney disease. In fact, somewhere between 50% of children who present with kidney disease and about 10% of adults who present with kidney disease have a genetic basis to their kidney disease. And in many cases, in the childhood kidney diseases, we really don't understand the genetic basis. And so in collaboration with the clinical geneticists and the nephrologists that are treating these patients, we're actually performing whole exome sequencing of patients. But frequently we'll find a novel mutation and we really don't know what that mutation is doing or whether it's causing a disease or not. With this strategy, we can actually take the patient's fibroblast, we can use CRISPR-Cas9 gene editing to fix a mutation that we may have identified during whole genome sequencing, and then we can make both a patient line, which has the mutations that occur in the patient, and a corrected patient line. And if we've actually corrected the right gene, we should be able to see a rescue. And so we've used these lines to actually build, it's actually not moving on, build kidney organoids from patient's line. This is the patient at the top, and the patient itself that's had a CRISPR gene-edited correction at the bottom. And we're looking at very high resolution at the individual epithelial cells in the nephron, and it's a bit hard to see here, but the red structures are the primary cilia of the epithelium, and this patient has a mutation in a cilia protein. And at the top, the cilia are clubbed. They have these big red blobs at the top, and at the bottom, the cilia are long and straight. And you can see that we can reduce the clubbing defect from 60% of the cells down to 10% of the cells by fixing one base pair change within that patient. So this is a very, we think, powerful way of actually proving that a particular gene is involved. So aside from disease modeling into the future, we are actually working to collect families in which there is heritable kidney disease and use this as a model for validating genes, but we can also then use drug screening approaches on organoids to see if we can actually find proteins that may assist the phenotype of those patients. In the longer term, we hope to be able to scale up to make much larger numbers of cells to start bioprinting new tissue, or taking cells and delivering them back into the patient for stem cell therapy, or even seeding cells back into decellularized organs in scaffolds to ultimately build a final organ. And I'll stop there.
Okay, thanks, Melissa. So we're done with our presentations, and you can ask any question that you might have. And we'll try to answer, yeah.
My name is Sharon Begley, and I'm from STAT. This is a question for Dr. Knoblich. Could you say a little bit more, please, about the biocompatible fibers that you mentioned, what they're made of, at what point in the development of the organoid you put them in, and how you think they act so that you don't get the organoid version of the messed up car that you should?
Okay, so the fibers are made in a very, at the moment, made in a quite a low-tech way. So they're made of a material that's called PLGA. That's a copolymer from lactic and glycolic acid, and this is the same material that's actually used for in surgery for the sutures. In this case, we use dental sutures because they are very rapidly biodegradable, and we add them at this very first step. So the very first step of our procedure, and also many other organoid procedures, is that you dissociate embryonic stem cells and then you aggregate them into what is called the embryoid body. And that embryoid body, if you don't do anything to it, is going to be round. And the three germ layers form in a way so that you have the ectoderm on the outside. And if you think about it, a ball is actually the worst way to get a lot of ectoderm, because the surface to volume ratio is the lowest for a ball. And this is how we think this fiber acts, because when you add the fiber, then the embryonic stem the pluripotent stem cells will form an elongated embryoid body. And we feel that this is how the variability of the organoid system is reduced. Ultimately, of course, I mean, we've tried a lot of different materials. For some reason, it only works with this one. And at the moment, we're working together with bioengineering groups to actually generate this from material that can be modified and coupled to certain chemicals so that we can induce a body axis.
My name is Tony. I'm a reporter at MIT's magazine Technology Review. I came late, so I didn't see it. Could you give a definition of organoid? What is the definition, and can you define it? What is it a blob? What is an organoid?
Yeah, so actually I think the various reviews that we've written, we try to come up with a definition of the organoid, but you probably might better define it than...
I actually have it. It should be on
Yeah?
the stick. So there's a...
So
I
maybe,
think
while Jürgen is looking for this, so the term organoid has been used in the 30, 40, 50 years ago for structures where you start with a tissue, you basically turn it into cell suspension, and you let them reform a tissue. That was the original term of an organoid. So they didn't involve stem cells, didn't involve culturing in the lab, didn't involve expansion of cells. So I think then the term wasn't used for 30 years. And about maybe the mid-2000s, Sasai was, in Japan, was one of the people who started this. So it is a different approach, a different use of the word. But Jürgen will give you now the definition that we think is that we all agree on.
Well, I mean, being German, we always need to have clear terminology. So we define it as a collection of organ-specific cell types that develops from stem cells or organ progenitors and self-organizes through cell sorting and spatially restricted lineage commitment. Sounds very complicated. I mean, basically, the elements of an organoid stands for like an organ. And in the broadest sense, it is a tissue culture that resembles an organ. And I think the minimal requirement is that it has some resemblance of the three-dimensional organization of an organ. So the cells arrange in the same three-dimensional way as they do in this organ. That it recapitulates some aspects of the physiology of that organ, like in the gut, it's the transport across the cells. In the brain, it's electrical activity or cell migration. In the kidney, it's nephron function and things. So this is the minimal definition. I think where different people in the field have different opinions is whether they need to come from stem cells. I feel that's included in the definition, yeah.
Yeah, maybe to separate this from what has been done with embryonic stem cell and iPS cells in the past, is there the efforts have been to make one type of cell, so make liver cells or make a certain type of stem cell or certain sort of kidney cells or brain cells. This was actually found more or less by chance, at least in my lab, but also in other labs, that stem cells have the capacity to do much more. So you can instruct them to make one cell type, and it will be a very monomorphic structure. But apparently, if you let them do what they do best, they will actually make many cell types, and they put them in the right positions to be a functional, at least part of a functional organ. So I think that has been the surprise. There's a lot of, so we don't really understand these processes, how exactly these cells are, all these cell types are made, how they sit in the right places, how they then connect to each other. We don't know that, but that's how nature does it, and that's what we mimic in the lab. Is that a?
Can I ask a follow-up question?
Yeah, sure.
Is it a human embryo, which except for this last one that it has to look like an organ, is a human embryo meet the definition of organoid?
So yesterday we had a session on organoid technology, and there was a fourth definition of organoids. They're called gastruloids. So an early embryo goes from a fertilized oocyte through very rapid stages. It becomes a blastula, and then it becomes a gastrula. The gastrula, so it basically it's a cyst, but then there's an indentation, and this starts forming what slowly forms into a real mammal or a real human being. That stage is called the gastrula, and that's the most important part of developmental biology of an embryo, of a human embryo, mouse embryo. And so it becomes possible now to actually, and there's 2 or 3 different cell types, different stem cells that make up a human embryo, to actually in the lab synthetically put these together, and they will go through the motions of starting to form a real human embryo. Now, there's of course lots of ethics and lots of questions to ask. Should we investigate this? How should we do this? So I think the research there is slow because we want to just be sure that what's being done in the various countries around the world really fits with the ethics standards of doing this kind of research. But that is, so you can actually, so we know, it hasn't been done yet, we know it should be possible to build a human embryo starting from the various stem cells rather than starting from a fertilized oocyte. Now you can probably think about the consequences that this will have, and one important part of the ICCR is actually to have a discussion before we start doing these real experiments. Now, do we want to do this?
Directly to the question, can we have hundreds of spinning bats growing people? Is that the ultimate? Once you have, if you can make a synthetic embryo, is anything to stop you from?
Can I make a point there? You actually asked the question, is a human embryo an organoid? And I would say no, because it's actually a perfect structure that will reliably produce everything. It's not a dissociation and reassociation of cells that makes a model of an organ. And most of our protocols for making a particular type of organoid do not pass through an identical stage that an early embryo does. They go towards particular cell types and they make complex structures, but they're not going through exactly what a human developing embryo does. It's an artifact in culture, and so we are not recreating a perfect brain or a perfect kidney. We're actually creating from cells in a dish a model that resembles a variety of different individual tissues.
If I may add on to this, I think when we come to these ethical discussions and can we generate an entire organ, like an entire brain, right? I think what Melissa said is very important. We are not precisely recapitulating development. We are actually doing, I think the kind of biology that we are recapitulating much more is that of a teratoma. So a teratoma is an ovarian tumor that arises from pluripotent stem cells, and teratoma is Greek for something that looks completely ugly or looks horrible, and that's what it is. A teratoma is a tumor that has various organs in it. There's teeth growing, there's hair growing and things, and you also find brain structures in this teratoma. And I think it is this kind of self-organizing principle that we are trying to recapitulate. And so in any teratoma, you have structures like this. We just try to make them a little bit better.
But I guess you're probing sort of the extreme limits of this technology. And it is, I mean, I would say, maybe I have a slightly different view, but I would say we have to address the fact that actually we do a lot of the things that happen during normal development of humans in bits and pieces, but in essence, if you look at what would happen at the far end of this whole process, we probably understand the entire process, and we could rebuild it. So it's extremely essential that we have the discussion. The better we get at this, do we want to do this? How do we want to do this? How do we want to regulate this, et cetera, et cetera. But I think, yeah, that's I guess your job to probe, you know, the extremes of this.
You're defining organoid, part of the definition is the limits that you encounter in the laboratory that's not normal. But if you overcame those limits, that would no longer be part.
Yeah, well,
I'm asking about the synthetic embryo. If you had this embryo, what would there be anything technically? Could you just
So technically, it is clear that an embryo can only grow inside a pregnant woman, right, in the uterus, and the next stage would actually be not a free-growing small thing, but would be what do you call this in English? An embedded embryo. So that's where it stops. IVF is now 3 days, 4 or 5 days in culture. With these gastruloids, it's a few days more, but that's about it. But my personal opinion is actually biology is usually, when we understand it, it's not extremely complicated, and we can always understand it. And in principle, eventually we can do what nature can. So to ask the questions, do we want to do this, and how do we want to do this, is extremely important. Because I think in the end we can. And maybe we have lots of practical complications, you don't want to hold the discussion. I think we have to address this now.
I mean, I should probably also say it would not be legal in most countries in the world. So there is already a legal limitation for gastroids. Gastroids can be cultured for 14 days, and that's it. Actually, this 14-day limit is being reached right now. There is a very intense debate about whether the 14-day limit is still up to date, or whether one should extend it further. But in essentially every country in the world, doing the experiment that you were just mentioning, generating an embryo in vitro in culture, would already be illegal.
Beyond the 14 days.
Beyond the 14 days.
But
So that's an entire embryo, right? Definitely.
Yes, definitely.
Definitely. Definitely. Yes.
We have time for maybe one more question. I wanted to see.
Thank you for exciting presentation. I'm Noriaki from staff writer for working for Japanese newspaper. And can I ask 3 questions? First question is, as the present organoids lacks red blood cells and nerves, I think. So is it a program for regenerative medicine?
Yeah, so let me first make one side remark. A lot of the technology actually comes from Japan. I think Shinya Yamanaka came up with the iPS cells. Toshiro Sato in my lab was back in Tokyo now, did the gut organoid. So I think the stem cell world owes a lot to Japan. People are, so now that we can have the sort of the most minimal version of organs, be it brain or lung or gut, scientists are now trying to add more layers of complexity. And actually, again, nature knows how to do this. So we don't really know how to be add the cells, but Jim Wells, for instance, had a very nice paper in my field where he grows from the iPS cells, he grows gut, and then he makes what's called neural crest in a different protocol from iPS cells, and these are the cells that make the nerves for the gut. And when he combines both, he actually makes a more complete gut. So now you have the inner lining, there's some muscle, but also the nerves now grow inside, and you can start seeing that they produce peristaltic waves. Now, we don't know, or Jim doesn't know exactly, what all the processes are that make this happen, but it's really surprising as a biologist to see that this happens in a dish. We just add 2 cell types, a bit like what Jürgen was saying about the 2 parts of the brain, you put them together and then nature takes the next multiple steps, which we don't understand, but we see it happen. And actually it's surprising how robust these processes are. So I know other people are trying to integrate blood vessels, then you would have to have circulation through the blood vessels, some blood needs to go through, but nature helps us a lot here. I mean, the cells know much better than we how to build these structures.
And second question is, there are two types of organoids. One is generated from pluripotent stem
Yeah.
cells, and another from adult stem cells. So, I want to know the impact of the discovery of human iPS to adult stem cells research. Is there any impact?
I mean, is there a link between the two? Or what?
And
You're asking for the impact.
Oh yeah, yeah. So I think they really are complementary. We're getting a little nervous in the back, but it's really complementary technology. So some things you can only do with adult stem cells. So I think particularly the cancers, if you want to grow the cancers or acquired, I mean, you have to start from the adult tissue. Tissues like brain that only form once and have a very hard time repair, you can only start from iPS cells, or complex structures like with Melissa, the kidney. An adult stem cell will not form an entire kidney. It will form parts of the kidney, but not the complete structure. But then there's also a lot of overlap, like gut or lung. You can form from iPS cells, but you can also form it from adult stem cells. So I think they are overlapping technologies where the iPS cells have some very clear advantages and the adult stem cells have some other clear advantages. And I think this is developing as a field, so we see different labs taking different approaches, and in the end it might even integrate.
I have a third question, but this is enough. Thank you.
Okay, I think we have to end our session here. Thanks very much for your very active participation, and thanks to the people behind the table.